Welcome, everyone, to this comprehensive explainer. Today, we're embarking on a truly fascinating and exhaustive biochemical journey. We're going to examine one of the most elegant metabolic pathways in all of biology, protein degradation. We'll literally be tracking a single protein from the exact moment it's ingested all the way down to the final excretion of its nitrogen waste. So let's settle in and take this step by step. We've got 5 distinct phases on our roadmap today. First up is protein digestion, followed by the ubiquitin proteasome system. Then we'll move into amino acid transamination, oxidative deamination, and finally, we'll wrap up at our ultimate destination, the urea cycle. All right, let's jump right into phase 1, protein digestion. This is where the breakdown of macromolecules begins. The whole breakdown process kicks off any pretty hostile environment, the stomach. The secretion of hydrochloric acid drops the pH drastically. Now, this highly acidic environment does 2 things. It acts as a defense mechanism against any ingested bacteria, but more importantly for us, it denatures the complex, folded structures of dietary proteins. In this acidic cauldron, an inactive enzyme, a zymogen called pepsinogen, undergoes autoproteolysis. It essentially activates itself, converting into active pepsin. As the stomach contents empty into the intestine, the environment has to shift. The hormone secretin signals the pancreas to pump out bicarbonate, which neutralizes all that harsh stomach acid. Once things are neutralized, a whole new wave of pancreatic zymogens enters the scene. Trypsinogen converts to trypsin, which then activates chymotrypsin. Together, they relentlessly cleave those long polypeptide chains into much shorter peptides. Finally, enzymes like carboxypeptidases A and B go to work, snipping off amino acids one by one from the C-terminal end. If we map this out anatomically, starting up in the stomach lining, you have parietal cells secreting the hydrochloric acid and chief cells secreting pepsinogen. That localized, highly acidic region is ground zero for denaturation. Following the digestive tract downward, we hit the pancreas and the small intestine. The pancreas duct is absolutely crucial here, releasing bicarbonate to buffer the pH back up to around 7 alongside vital zymogen granules packed with our inactive proteases. Finally, down in the villi of the small intestine, we reach the end of the line for digestion. This is where our completely freed, fully digested amino acids are absorbed into the epithelial cells, ready to be shipped off to the liver. Moving right along to phase 2, the ubiquitin proteasome system. So, we just covered exogenous dietary proteins, but how does a cell manage its own internal, endogenous protein turnover? Well, we have 2 main systems at play. On one hand, there's the lysosomal mechanism. Think of this as bulk, largely non-selective degradation. It relies on lysosomes, which are highly acidic organelles with a pH hovering around 4 or 5. Cellular material gets swept up via endocytosis or autophagy, fuses with the lysosome, and robust hydrolases just break everything down. On the other hand, we have the cytosolic mechanism. This one is highly precise, incredibly selective, and ATP-dependent. It specifically targets short-lived regulatory proteins or proteins that have misfolded. And to achieve this level of surgical precision, the cell relies heavily on the ubiquitin proteasome system. To really grasp how this selective degradation works, we need to understand the chemical glue that holds the destruction tag together, the isopeptide bond. Ubiquitin is this tiny, highly conserved protein, just 76 amino acids long. When a cell needs to mark a target protein for death, it attaches a chain of these ubiquitin molecules to it. The links forming this chain are isopeptide bonds. Specifically, it's a covalent link formed between the carboxyl group of the C-terminal glycine on one ubiquitin and the epsilon amine group of a lysine residue on another ubiquitin. Nailing down this chemical bond is vital because it's literally the foundation of the polyubiquitin destruction tag. So, how exactly does this tag get attached? It takes a beautifully orchestrated 3-enzyme relay cascade. Step 1 involves enzyme E1, the ubiquitin activating enzyme. E1 burns energy, consuming ATP, to activate the ubiquitin molecule. Step 2 brings in enzyme E2, the ubiquitin conjugating enzyme, which basically accepts that activated ubiquitin right from E1. Finally, step 3 relies on enzyme E3, the ubiquitin ligase. You can think of E3 as the matchmaker. It brings the E2 ubiquitin complex and the specific target protein together, allowing the ubiquitin to be transferred and bound directly to the doomed protein. If we trace the actual pathway of these enzymes, ubiquitin gets activated first by E1 using ATP. This forms a high-energy thioester bond between E1 and the ubiquitin. Then E2 steps in and takes handoff of the ubiquitin. Finally, E3 facilitates the ultimate transfer from E2 straight onto a lysine residue of the target protein. Now, a crucial point here is that this process repeats. It's these repeating cycles that build up a long polyubiquitin chain, firmly sealing the protein's fate. Once a protein is lugging around that heavy polyubiquitin chain, it gets recognized and dragged to a massive molecular shredder, the 26S proteasome. At its center is the 20S core. It's made of 4 stacked rings, 2 outer alpha rings and 2 inner beta rings. It's actually inside those inner beta rings where the real proteolytic chopping happens. But a fully folded protein can't just squeeze into that narrow cylinder. That's why we have the 19S regulatory caps on both ends. These caps are packed with ATPase subunits. They recognize the ubiquitin tag, forcefully unfold the target protein, and feed it right down into the 20S core, where it gets sliced up into short, reusable peptides.
Phase 3 brings us to amino acid transamination.
Now that we've seen how proteins are broken down into individual amino acids, we are entering the metabolic phase. The big challenge here is safety. To safely catabolize an amino acid's carbon skeleton for energy, we absolutely have to remove its alpha amino group first. As long as an amino acid holds on to that amino group, it's pretty much protected from further metabolic breakdown. In a transamination reaction, an L-amino acid acts as the donor. An enzyme, an aminotransferase, plucks off that amino group and hands it over to an acceptor. Almost every single time, that acceptor is a molecule called alpha-ketoglutarate. By accepting the amino group, alpha-ketoglutarate transforms into L-glutamate. Simultaneously, the original amino acid, now stripped of its nitrogen, becomes an alpha-keto acid. This funneling of amino groups to create glutamate is an essential metabolic bottleneck. The enzymatic reality behind this transamination is wonderfully complex. It follows a 2-part ping-pong bye-bye mechanism, relying heavily on a vitamin B6 derivative acting as a cofactor, pyridoxal phosphate, or PLP for short. First, the incoming amino acid reacts with the aldehyde group of PLP, forming a covalent intermediate called a Schiff base. Second, the amino group is left behind on the cofactor. The original carbon skeleton floats away as an alpha-keto acid, and our PLP has now morphed into pyridoxamine phosphate, or POMP. In the third step, the second half of the ping-pong reaction begins. Our acceptor, alpha-ketoglutarate, arrives and reacts with the PMP. Finally, in step 4, the amino group transfers onto the alpha-ketoglutarate, forming glutamate. This brilliant maneuver restores the cofactor back to its original PLP state, primed and ready for another round. Let's move to phase 4, oxidative deamination. So, through transamination, we've successfully funneled all this cellular nitrogen into a massive pool of glutamate. Now we have to free that trapped nitrogen so the body can actually excrete it. But this raises a really fundamental structural question. Out of all the molecules in the cell, why do we specifically produce glutamate during transamination? Why funnel all those amino groups onto alpha-ketoglutarate in the first place? The answer comes down to enzyme specificity. Glutamate is uniquely capable of undergoing rapid oxidative deamination. Other amino acids just don't have an equivalent, highly active enzyme to efficiently strip away their amino groups in humans. Glutamate is the chosen carrier. This critical step is driven by the enzyme glutamate dehydrogenase. Glutamate enters the reaction, and the enzyme uses NAD+ or NADP+ as a cofactor, reducing it in the process. As this happens, the amino group is cleaved off, liberating itself as a free ammonium ion and perfectly regenerating our old friend alpha-ketoglutarate. What's fascinating is how tightly regulated this reaction is by cellular energy levels. When cellular ATP is low, this reaction is strongly stimulated. It ramps up production of alpha-ketoglutarate, which can immediately drop into the Krebs cycle to generate more much-needed energy. And that brings us to phase 5, our final stop, the urea cycle. We've broken down the protein, we've removed the amino group via transamination, and we've liberated it as highly toxic free ammonia through oxidative deamination. But how does this toxic compound finally get eliminated from the human body? Free ammonia in the blood is incredibly dangerous, particularly to the central nervous system. Different animals handle this differently. Aquatic animals can just flush it right into the water, and birds convert it to uric acid. Humans, however, have to convert and excrete this nitrogen almost entirely as a highly soluble, non-toxic compound. That compound is urea. The urea cycle is the metabolic pathway that pulls off this remarkable feat, and it actually spans 2 distinct cellular compartments. The cycle initiates deep inside the mitochondrial matrix. Here, the freshly generated free ammonia combines with bicarbonate. This burns 2 molecules of ATP to form a high-energy intermediate called carbamoyl phosphate. This is a critical, irreversible step.
Carbamoyl phosphate then joins with ornithine to form citrulline. The citrulline is shuttled out of the mitochondria and into the cytosol. There, it meets a second source of nitrogen, the amino acid aspartate. They condense together into a massive molecule called arginine succinate, which is then cleaved. Fumarate breaks off to join the Krebs cycle, leaving behind arginine. Finally, the enzyme arginase cleaves the arginine, producing our end product, urea, which gets excreted by the kidneys, while beautifully regenerating ornithine to keep the cycle turning. To really lock this in, let's map out the exact atomic origins of that final urea molecule. Urea is a beautifully symmetrical molecule containing 2 nitrogen atoms, 1 carbon and 1 oxygen. So where did they all come from? Well, the first nitrogen atom comes directly from the free ammonia we produce during the oxidative deamination of glutamate inside the mitochondria. The second nitrogen atom comes entirely from the amino acid aspartate out in the cytosol, which, by the way, got its own nitrogen via the transamination of oxaloacetate. Finally, those central carbon and oxygen atoms derive from bicarbonate, which originates from cellular CO2. Every single atom is accounted for, meticulously tracked from start to finish. And just like that, we reached the end of our journey, the balance of life from a single ingested protein down to a single molecule of urea safely excreted. We've methodically followed the acidic denaturation in the stomach, the highly selective destruction by the proteasome, the elegant ping-pong transfer of transamination, the liberation of ammonia, and finally, its safe packaging within the urea cycle. It truly is a staggering display of metabolic precision.
Before we close out this explainer, I want to leave you with a final thought to chew on. We've spent this entire time obsessively tracking the nitrogen. But what about the carbon skeletons left behind once those amino groups were stripped away? What other metabolic pathways intersect with the fate of those remaining alpha-keto acids? Will they become glucose through gluconeogenesis, or will they transform into ketone bodies? The answers to those questions sit right at the heart of cellular energy regulation. I hope that leaves you eager and curious for our next deep dive into biochemistry. Thanks for joining me.